Welcome, everyone. My name is Ellie Juarez. I'm the Associate Director for Global Segment Marketing here at Oxford Nanopore. And I'm excited to welcome you all today to this online session where we'll be looking at adaptive sampling, which is a unique software-based target enrichment method from Oxford Nanopore Technologies. After all the talks are over, we'll have a live Q&A session. So add your questions to the speakers today to the Q&A box, which you'll find at the right of your screen. You can also upvote the questions you would most like answered, and we will try to get through as many as we can. This webinar is being recorded and will be available on demand after the broadcast. And so with that, I'd like to get started and maybe just tell you a little bit, set the stage for my colleague Miguel Reich, who will be joining me in just a few slides. And so I'd like to get started just by telling you a little bit about Oxford Nanopore. So the grand majority of our technology is really powered by this small blue protein you see here embedded in this synthetic membrane. And our technology is really based on a library prep solution. We've got a suite of library prep solutions, but an important component of that is attaching the motor protein that will bring the nucleic acid over to the nanopore that is embedded in the synthetic membrane. That then threads the nucleic acid through the membrane. And as we run an electrical current through the membrane, you get these sort of squiggly lines that you see here on the screen. And those squiggly lines, we are able then to look and discern an A is slightly different than a T, is slightly different than a G, and a C, importantly, is slightly different than a methylated C, giving us a multi-omic view of the genome. All of this is done in real time for rapid insights. You can do native DNA or RNA sequencing to get the epigenome. Any fragment links, because we're really just pulling a string through the protein. And we're powered by end-to-end workflows that I'll talk about in just a second a bit further. And this nanopore is the basis of a variety two flow cells that we have in our suite. Those are the MinION and PromethION flow cells. The MinION flow cells have a lower capacity, but still are powered by the exact same technology, versus the PromethION flow cells just have more of those nanopores in them and are therefore able to create a larger output of sequencing data. Importantly, those two flow cells then fit into our suite of devices that really cross a large continuum from personal sequencers like our MinION that's smaller than a cell phone to scale-up sequencers like our P2i that has an integrated compute and can hold two of these PromethION flow cells, and then our high-output sequencers for population-based studies that hold 24 PromethION flow cells that can be run independently of one another, giving you a momentous amount of flexibility to meet your high throughput sequencing needs. And all of this is then wrapped into a fully supportive, intuitive solution in our analysis using the Epi2Me platform. So this is an intuitive interface, a point and click GUI, if you will, with pre-configured workflows. And I'll talk a little bit later, as will my colleague, about some of these workflows. There's standard output files like VCF and BAM files, and you can run it locally or in the cloud. You can also have command line access and easy integration, providing kind of a flexible suite of things. Choose your own adventure, really depending on your compute needs and compute capabilities for any individual assay. And so Epi2Me, from an end-to-end workflow, you get a DNA or RNA that is sequenced. Then the files are written locally. You analyze with Epi2Me. And then you choose between running the analysis in the cloud or on-prem, and that really just depends both on your computing needs and capabilities, as well as any regulatory body concerns that you may have. Either way, you get files and outputs that are generated. You can also get, if you run it in the cloud, you can download those results then onto your local system. So really providing a flexibility depending on both your regulatory as well as computational needs, all in this point and click GUI. But again, we try to not be black box here at Nanopore, so we also have command line capabilities for you. And so, really, that's just setting the stage for what Nanopore and kind of our capabilities. But we'll be taking a much, much deeper dive into adaptive sampling today, which is a modality for target enrichment. And really, currently on market, there are several target enrichment workflows. There are amplicon-based, these are primer-based amplification of the targets of interest, tend to be smaller panels, have relatively quick workflows, anywhere between 2.5 and 8 hours, and have a limited amount of variant types that they can produce. Conversely, you have hybridization workflows. These are probe-based, primarily biotinylated probe-based pull downs for your regions of interest. These do tend to be much larger panels in the 500, even 750 genes, but they are quite long, often cumbersome workflows that can take 2 to 3 days to complete. And then today we'll be talking about this technology that really is unique to Nanopore, adaptive sampling, which is a software-based on-sequencer target enrichment methodology that has a lot of flexibility in both the size of the targets and the number of genes that are targeted, a very quick workflow, and really provides this multi-omic view of the genome with single nucleotide variants, copy number variations, and methylation as well, using that direct DNA sequencing. And so now I'd like to hand it off to my colleague, Miguel Reich, a development scientist and our adaptive sampling expert here at Oxford Nanopore Technologies. Welcome, Miguel.
Thank you, Ellie. Hello, everyone. My name is Miguel Reis. I'm the lead scientist in the development of adaptive sampling at ONT, and I'm here today to explain to you a bit better about the intricacies of adaptive sampling. So I would like to start by quickly reviewing the ONT sequencing system itself. The way that the DNA is sequenced with our platform is that we take strands in a sample and they get attached to a pore, and as they go through the pore, the signal is collected in real time regarding whatever bases are going through the pore at any given moment. Adaptive sampling takes advantage of this because this allows us to read the strand as it is going through the pore in real time. With this, we can take the signal from this particular strand, base call it, align it to a reference, and make a decision of whether do we want this strand or not. If we do want the strand, we do nothing. We just let the strand keep getting sequenced and reaching its natural end. However, if we realize that this strand is not of interest, we can reverse the polarity on the pore and reject this strand back to the chamber where it came from, allowing another strand to be captured, in this way accelerating the sample at which we collect information about the sampling rate at which we collect information regarding our sample. This results in enrichment, and it results in an output that is heavily skewed towards coverage on on-target regions. As you can see from the picture, we're going to end up getting a lot more longer reads on our on-target regions, but we will still also get very short reads on our off-target. This will provide us a backbone of low-pass sequencing on all our off-target regions, and this can be very useful for CNV analysis or for creating a methylome backbone of the whole complete genome at no cost to your on-target enrichment sequencing. This technique is also very simple to prepare because there's literally no difference in the protocol for a library prep between a whole genome sequencing approach and an adaptive sampling approach. It's PCR free, and it's also more cost effective than having to perform hybridization capture or having to buy probes to target to each one of your targets. It has an extra advantage, which is the fact that we can also modify our targets on the fly and very easily, so you don't need to wait for new probes to try a new enrichment panel. You can easily add it to your bat file and try another panel without much hassle. So this is all done and provided by the use of a combination of a reference and a bat file. So you might already be familiar with reference files, in particular FASTA format, where you have a description of your complete sequence of the sample that we're providing. And this sequence will describe every single strand and base that you may find in your sample. The other side of this is the bed file, in which we specify out of this reference what are the regions that we want to target. And this is done by simply taking the first column of the bed file and specifying the tag used by the chromosome in the reference, using the second column to specify the initial coordinate and the last column to specify the last coordinate. So between these coordinates lie the bases related to the region of interest that you want to enrich for. And as you may have understood, each one of these lines will specify a different target that we are interested in enriching for. So how does all of this target happening in the system? So if you imagine that this blue line here represents your FASTA, your reference file, containing all of the sequence present in your sample. In orange, we marked regions of interest that we would like to enrich. So these are the regions that would be marked in your bed file. In brown and green, I depicted what a strand would look like aligned to this particular FASTA and bed regions. In brown, I marked the beginning of the strand. So this would be what we call the initial chunk, which is the first 400 bases of the read that go through the pore. We take these and we analyze these against the FASTA and the bed file to check where they fall and if they fall within a region of interest. So wherever this brown chunk falls and aligns in is what we take as a consideration to make a decision whether we're going to accept or reject. So in example A, this initial chunk falls within the region of interest. We will accept this strand and we will sequence this strand. In example B, this first initial chunk that goes through the strand, through the pore, does not fall within our region of interest. It falls in a flanking region, and therefore it is not in the region of interest. We will not be accepting this strand. Nevertheless, as you may have imagined by now, this strand, would it have been sequenced, would have actually ended up reaching our region of interest. So this strand would have been of interest to us, but we were not able to capture it by simply targeting this way. So what we can do is we add buffer regions to our target of to our regions of interest. So if in this picture you can see in red, I added an extra region flanking my target region, and these we call them buffer regions. And in the pink square you can see this is the total target that we are actually practically targeting with adaptive sampling. And what this does is that if an initial chunk of the read falls within this region, instead of getting rejected as before, it gets accepted because Minnow makes no distinction between what we set as a buffer or as a target. For Minnow, everything inside this pink bracket is region of interest. And therefore, we will accept this trend, and this trend will then keep being sequenced until it reaches the region of interest and provides us with the coverage on target that we want from an enrichment methodology. Have these buffers been defined correctly, we can see that reads that don't extend into our region of interest have their initial chunk fall just outside the buffer region, which will make these reads not accepted and rejected, and it allows us to not carry over too many false positive reads in the sequencing. So it is a very important consideration that we think about how long or how short these buffers might be. In situation A, we have an ideal sized buffer, and we have the initial chunk falling on the buffer, and the strand gets accepted. it is sequenced, and there is a considerable amount of sequence of the strand that is actually on target and of value. On situation B, we have a buffer that is too short for this read, and even though the read did eventually or would eventually reach our region of interest, it ends up getting rejected because the initial chunk falls outside the region of interest and the buffer that we defined. Contrary to that, we also have situation C, where we defined a buffer that is too large for our strand. So the initial chunk does fall within the buffer. The strand gets accepted and extended, but unfortunately it never reaches our region of interest, making it a complete waste of sequencing time. And this is also a situation we want to avoid. Another point important to consider is, besides the buffer, what our library size is. Because if the library, well, using a technique like ours, we have the tendency to want long reads because we can just do them, and the platform does not care about sequencing longer reads related to shorter reads. So why not just have a longer library that's always of use? In the case of targeting, that is not particularly the case, because as we can see here, if we have a smaller strand and two equally sized targets, this strand will get accepted, it will get sequenced, and all of the strand that we sequence is of interest here. Now, in situation B, we have a much longer strand, which would it have been accepted in the case of having ideal buffers in here, it would have extended into our region of interest, and it would have provided us with a complete coverage of the region and a 1x coverage complete across our region of interest. However, this strand, for the sake of argument, takes approximately 10 seconds to get sequenced. And comparatively, the strand in situation B would take 40 to 50 seconds getting sequenced, out of which only a small amount of it would actually be region of interest and on target. What this does is that it wastes a lot of time sequencing when it provides a very small amount of the strand only as on target. This makes for a lot of inefficient sequencing time that could have been used to sample faster and to get more reads that fall on target. So this is to exemplify that having longer libraries is not always good for a targeting approach. And speaking of buffers, we should take a look at what should an ideal size or what would an ideal size of a buffer look like. And it is a complicated question which has a lot of variables to pull, but as a rule of thumb, I like saying that an N10 of the read length distribution of the library we're using is an ideal value. So if you look at the read length distribution here, picking something like the value of the N10 of this library would be an ideal value to add to either side of the targets in our targeting buffer, in our targeting bed file. I also want to call your attention to how this affects the amount we're targeting on the bed file. I have two examples here. Example 1, we have a panel which contains 200 targets. In example 2, we have a panel that contains 18,000 targets. Now, I chose ends of the spectrum for the purpose of an exaggeration, but I want to show you that in example 1, this panel targets 0.5% of the sample, and with buffer at 14 kb, it ends up targeting 0.62. So the increase in amount targeted of the sample after adding the buffer is minimal. On example 2, the targets only themselves are already bigger. They target 4.5% of the sample. And after adding buffer to 18,000 targets, we end up targeting 15.28% of the sample. And this is above our recommended threshold of 10%. Ideally, you do not want to be targeting more than 10% of your sample to try and maximize the amount of time you spend sampling and looking for your regions of interest. This will maximize your enrichment. Another consideration regarding the library length is how it affects the decay in pores. So if we look at mux scans from a library of N50 equals 6 kb in a library where the N50 is 20 kb, we can see that the depletion of time in the same period of 20 hours is much higher in a library of 20 kb N50 comparatively to the 6 kb. This means that the 6 kb library will run for longer. It will keep your flow cell healthier and alive for longer, allowing you to collect more data on target. This also avoids more frequent flushes. And thinking back to our targeting approach, if your targets are not very big, having a bigger library could, on top of all of this, be wasting sequencing time by having strands that are much larger than the single targets described in the bed file. Lastly, regarding library, it's also important to think how a longer library affects your pore occupancy. Pore occupancy is a measure of how much pores are spending in sequencing versus in open pore or in a state where they don't have a strand on them. So we want to maximize pore occupancy because that means that our flow cell spends the most time in sequencing and you end up acquiring the biggest amount of data. This is also important regarding pore depletion, as a sequencing run with a high amount of open pore may result in the pores depleting faster. And as we can see here, a library with 84% pore occupancy, 91 and 99, we can see that the library in the run where we keep 99% pore occupancy has a much smoother decay in pore loss comparatively to the other two libraries. Thinking about how loading in library molarity works, we have a much harder time achieving high molarities necessary to keep the pore occupancy up if we are using a library that is much longer, because you would have to load a much higher mass to have the exact same molarity of a long library as you do of a short library. So often shearing your library into reasonable sizes can facilitate all of these aspects, allowing you to have a run that performs a lot better with much higher outputs, allowing you to reach the coverages that you need. So what do you need before you start a run? Well, you need a reference for your sample. You need a bed file which contains your regions of interest plus buffer, and this is the bed file that you will give to adaptive sampling to do the targeting. And then optionally, you can also use a bed file without buffer, which you will provide to the live sequencing base calling so that it can track your coverage more accurately. I say this is optional because you don't necessarily need to do live base calling and alignment. And even if you do, you could potentially use the same bed file with the buffer to track the coverage. It's just that it would be slightly more accurate if you use a targets only versus using the targets only and buffer for the live measuring of the coverage obtained. It's also important to consider whether you will want to flush your run or not. And if you do, you should have prepared a flow cell wash kit alongside with the number of libraries or loads of the same library that you will need after you have flushed your flow cell. Now I would like to quickly go into our Minnow software and show you how to set up an adaptive sampling run in our platform. So I would now like you to take you very quickly through the setup of an adaptive sampling run. Here we can just write adaptive sampling run. We select our flow cell and our sample. In this case, let's do an hereditary panel. Select our kit. And in here we have the options we can choose from. So let's turn on adaptive sampling. And in here we can select our reference file, our FASTA. On the menu below, we can upload our bed file. Here it is, targets plus buffer. and we can validate them to make sure that they are correct and they have no errors and that they play with each other. We also have the option to enrich, deplete, or base call on target only. So if you do turn on base call on target only, this is a new feature that allows you to rebase call only the on target reads, and this reduces the stress and load on the machine heavily. So this could allow you to use SUP base calling for your on target reads only on some devices with some configurations. This will, however, not rebase call your rejected reads, so they will still be saved as pod files if you choose to do so. They will just not be base called live, so you could base call them later if you would need your rejected reads. By clicking here, you can turn on live alignment, and you can see that it gets pre-populated with the same reference file and the same bed file. You can click here to modify the bed file in case you want to load one with targets only. This will make tracking coverage a bit more accurate, but it's not strictly necessary. And in here, you can modify the base calling model. Now, this base calling model is what I will describe later a bit better, but it's regarding live base calling reads. So because we have chosen base call on target only to be on, this base calling model is the base calling model that will be used to rebase call the on target reads only. So in this case, we have a single flow cell on a capable device, so we can use SUP live base calling for our on target reads. And that's pretty much it. You can choose other options if you want to, but our adaptive sampling run is ready to go. So I would like to take a quick look at two very important things that we saw in the Mino software. We will have in enrichment runs an adaptive sampling base caller, and we will have a live base caller. Hence why we also supply two sets of references in bed files. The adaptive sampling base caller is a fast base caller which aligns in short read mode, and it's just on by default and the user has no control over this. This is what we use to base call that first initial chunk and make a decision on whether we're going to keep your read or reject it. As the live base caller is the one that will rebase call your complete read with whatever model you choose and will align it. And this is something that the user is in complete control. You might not even have this on if you don't want to have your data live base called. This is completely up to the user to choose. The reason I bring this up is because it's important to understand that when doing an enrichment run on the device, you will be asking the device to do two sets of work on the run. And this is why these runs are often more heavier on the device, and we need to be a bit more careful about the amount of features that we turn on when using adaptive sampling and the number of flow cells that we're running per device, etc. Another thing I would like to call your attention to is how to understand if you are overloading the system. A very quick and easy way to see if an adaptive sampling run is performing all right is by looking at the N50 of the rejected reads. So if you go into the panel of the library length histogram, you will see, and if you click estimated and you hide the outliers, the run will show you this purple peak. This purple peak is the representation of the rejected reads. And this gives us a very good idea of how long the system is taking to respond to a read. So the faster we kick reads out of the port, the faster we are sampling, and this is a good measure of how healthy and how well the system is keeping up with the load we are asking it to work with. So if you have anything near 600 bases as a rejection peak for the adaptive sampling voltage reversal, you should be fine and you know that your system is working correctly. If at any given point you see that this number goes somewhere above 800 bases, you might have your system clogged and you might have too many flow cells or features turned on, and the run will not be enriching as much as it could have been if the system was performing correctly. Another interesting thing to look at is, and this is a bit panel dependent, is how much you're targeting. If you tick off the hide outliers, you will be able to see or focus on the region where you expect your library length to be. And it's a bit hard to see, but there's a smear of green that we see in this region. This corresponds to our accepted reads. And a balance between the peak of accepted reads versus the peak of adaptive sampling, it's something that it's always interesting to check to make sure you're targeting what you think you're targeting. So this is an example of a panel that is targeting 0.7% of the sample. And as you can see, by targeting 0.7% of the sample means we will only be accepting 0.7% of the reads. So that's less than 1% of the reads that go through the pore actually get accepted for sequencing. This results in a very thin smear of green, and this is the sort of ratio you should expect when you are targeting such low amounts. If at any point in your run the green amount is higher than the rejected amount, then your run might not be working properly, or you are somehow targeting way too much and you won't be getting high levels of coverage or enrichment as you might expect. Adaptive sampling is a feature enabled in all our devices. It's important to keep uh, consistent with our recommendations regarding what features and how many flow cells you can run in each one. We have this available in the community, but just know that it is a feature available for all our platforms in sequencing flow cells. Also, it is a feature that we allow for users to customize by themselves, as well as we provide some protocols and workflows that are predefined in which we optimized all of these conditions that I've been talking about so that you get the best out of the run for particular panels. These panels have end-to-end workflows and solutions. They have epitome workflows that analyze the results so that you get the complete set and experience from DNA extraction all the way to results. On the other end of the spectrum, you are also always welcome to tinkle with it and have your own bat file and play with it and play with your libraries to try and find optimal protocols for whatever panels you need to target. This is also enabled, and it's free for the user to try and experience. So as I mentioned, we have our own protocols, our own end-to-end workflows that we supply with, and these are the hereditary cancer one and the PGX workflow, and we also have the human variation workflow, which does some standard analysis on the outputs of the run. Now, the first two are adaptive sampling specific workflows, and we do have end-to-end protocols for these. However, you can always run any of these protocols or any of these epitome workflows with your outputs in case any of these solutions adapt into your particular needs. So the take-home messages from this presentation are mainly regarding library preparation and the input preparation that you need to do an adaptive sampling run. Most important thing is to make sure you have good quality DNA to start with, and then take into consideration what we talked about regarding DNA length and how to target and how to build a bed file and how to buffer the bed file appropriately to the DNA length that one has chosen. One consideration that we mentioned in the community as well is that adaptive sampling is a bit more hungry in terms of sample than whole genome sequencing. So we recommend 50 femtomoles of library as a minimum to have a decent run. The more you input, the better. So if you are able to provide 100 femtomoles of your sample, that will be better. But please just make sure that any time you start a sequencing run with adaptive sampling, you check your pore occupancy, and ideally you keep it above 90%. Also, have those wash kits ready in case you want to flush and reuse your flow cell. Make sure you prepare your FASTA and bed file, and that you have buffered your bed file correctly. And then 10 as a rule of thumb out of your library read length is a good measure for buffer. And when it comes to base calling, please follow the community guidelines as we are constantly updating these. But in most devices nowadays, you should be able to keep up with hack live base calling in most conditions. If you're not sure, then fast is always a safe choice. I would like to leave you here with some links to useful pages in our community. You have the adaptive sampling info sheet where you'll have all the information I've talked about today, plus some other deeper dives into some sections of it. We'll have the adaptive sampling best practices, which talks about library preparation and how library lengths and occupancy affects the run. We also went through. I'll leave the link here for the pharmacogenetics and the hereditary cancer panel protocol. So these are two end-to-end solutions that we are now supporting, and that you can get the end-to-end workflows from us. And with this, I hand over to my colleague Ellie. Thank you.
Thank you, Miguel, for that expert deep dive into all things adaptive sampling. Miguel will be back later in the broadcast to take part in a live Q&A. Now, I want to talk a bit about our out-of-the-box ready adaptive sampling suite of products and how our customers are using this customizable and flexible technology that Miguel just highlighted for critical research questions. So really the impetus of adaptive sampling, as I think Miguel has expertly showcased, is to really simplify target enrichment and accelerate both turnaround time via flexible sample volumes, as well as comprehensive data within a single assay that you can see with these end-to-end epitome workflows that Miguel has given a brief overview of. And so I thought I'd take a little bit of a deeper dive into some products that we have available today. And so first, I thought I'd start maybe with some of the limitations to our current testing. Target enrichment panels primarily today are done via short read NGS sequencing, and those have a cumbersome component of the workflow, primarily around library preparation and target enrichment, with high hands-on time. Panel content is often fixed. Adding and subtracting probes is quite difficult, versus Miguel has just really highlighted for us how easy it is to just customize the bat file with it using adaptive sampling to really fit for purpose needs of your panel. And updating the panel content can often be costly. You have to buy new probes. You have to revalidate those assays. Sequencing can take 24 hours, but also requires a large batch sizes, often getting a lot of samples in, which can really just increase turnaround time to results. And again, you've got some limitations for short reads themselves, which can really sort of show up in limitations in variant calling for things like structural variants. Intronic regions are often not covered with short read methodologies, and methylation because of the PCR necessary for most standard amplicon and hybridization workflows. You often require additional testing to get that second multi-omic modality. And so for the first product that we've tried to create to answer some of these limitations with our current on-market target enrichment products is the Oxford Nanopore Hereditary Cancer Panel. It's a simple workflow that allows for assay consolidation, again, getting this rich multi-omic data set. You've got less than 2 hours hands-on time in the library preparation step, a very quick, again, extraction, fragmentation of the DNA, and then just ligation of that motor protein I discussed at the very beginning that brings the nucleic acid over to the nanopore. You load and go sequencing. Because the enrichment happens on sequencer, it's really a set it and forget it sort of situation. And then importantly, using this end-to-end workflow that Miguel discussed earlier, the workflow hereditary cancer, you get 1 hour per sample worth of analysis, but that's giving you structural variants, these intronic regions. And I'll highlight a customer who's using a custom version of the hereditary cancer panel to look at deep intronic variants later in this talk. And then we have coupled this with interpretation and third party for any reporting that you might need downstream. And so this is our newest product that was launched on September 3rd. And we've been able to show some really exciting data about the usability of this panel. We were first wanting to test how well does the hereditary cancer panel with adaptive sampling perform in calling small variants like single nucleotide variants and indels. And here you can see that there are 9 variants across multiple Coriell samples in genes that are known to be inherited for hereditary cancer, for example, VHL, which is a gene in von Hippel-Lindau disease, BRCA2, which I think has been extensively studied, XPC, which is a gene in xeroderma pigmentosa. And importantly, not only were we able to find all 9 out of 9 variants, but we were able to find them in the appropriate variant allele frequencies here. And so zooming into one of these variants, the von Hippel-Lindau VHL mutation, we're able to show not only that deleterious 8 base pair deletion in VHL, but also given that multi-omic direct DNA sequencing view we can do of the genome, we were also able to show this hypomethylation of the VHL promoter and exon 1, which is denoted in blue here in the bottom figure. And that really kind of shows how you can layer on not only mutation information, but also methylation information to make the most comprehensive decision about the variants that you're seeing within this panel. Given our NERI technology and our capability to sequence really large stretches of the genome, we were also able to show that in the SeraCare control that has a BRCA1 and BRCA2 deletion, we're able to show, for example, this 23 kb deletion in BRCA2. And I really invite you to go look at our app note and our flyer. We'll have some QR codes at the end where you can find more information. This is just an example of a few mutations we were able to find with the hereditary cancer panel, but you can deep dive as well as look at some published data looking at the performance of this particular panel. Our next product I wanted to discuss is another adaptive sampling out-of-the-box solution. Again, it has that end-to-end workflow with the workflow PGX or pharmacogenomics. And this is a sample looking at that is able to provide high pseudogene homology resolution, spacing of dense and common variants, able to provide resolution of complex gene and pseudogene hybrids as well, and copy number gains and losses all in a single solution. Again, this is powered by adaptive sampling. The PGX panel, sort of by the numbers, has gene coverage of 375 genes, which covers about 10% of the genome. The read length along this panel is about 8 kb. You can multiplex samples. It's 4 samples per that Prometheon flow cell that I highlighted earlier in the talk. And the lab workflow, again, is simple and easy, half a day sample and library prep with 72 hours of sequencing. And again, I invite you to visit our pharmacogenomics product page to learn more, download the flyers and app notes to look at all of the data as well as looking at the published data sets. And so these are kind of 2 of our out-of-the-box products that have end-to-end workflows. But as Miguel highlighted earlier, the beauty of adaptive sampling is really its flexibility, your ability to change that bed file, that chromosome start-stop to fit whatever scientific needs you have. And so I thought I'd next highlight a few publications that have been doing using adaptive sampling in their customized workflows to answer some really critical questions in biology. And so this publication here is from a group out of UNC and in collaboration with St. Jude, where they were really looking for rapid classification of leukemia in pediatric samples. And what they were able to show is using their custom adaptive sampling workflow that they were able to get answers in 6 to 9 hours end to end. The sequencing was done on a P2 solo and using adaptive sampling and real-time base calling on Minnow, and they were able to get a karyotype in as little as 15 minutes and structural variants in 3 to 6 hours. So really get a very comprehensive view of these pediatric samples. We're getting a sample to answer in a really quick time can really make a difference. And so I invite you to scan the QR code at the bottom to read this publication and really see how our customers are leveraging adaptive sampling. And so here's an example of that copy number variation that they were able to assess. They did what's called a digital karyotype using that low pass whole genome view that you get of adaptive sampling. And you can see here in figure A, it's a pretty complex karyotype with lots of variation, with gains and losses of chromosomes, as well a slightly less complex, but still multiple changes to the genome in chromosome B. Importantly, not only are they able to get that copy number variation, but they're also able to get this rapid and cost-effective classification of more molecular diagnostic view of the genome. And so here you can see large complex rearrangements in DUX4, in IGH, and a large deletion in ERG, kind of like the one I showed you using the hereditary cancer panel. And again, I invite you to go read this paper that I think really highlights some of the powers and the flexibility of adaptive sampling. And so the next publication I wanted to highlight is from a group out of the University of Washington, Tom Walsh and Marie Claire King. And in this particular question, they wanted to leverage adaptive sampling to get questions for families who maybe didn't have answers before, or these Cancer X families. Families that, if you look at their pedigree, they have a very clear inheritance phenotype that is suggestive of an inherited gene across the family, but for which, using whole genome or whole exome and whole transcriptome, they had still been unable to find a causative gene that was really driving that inheritance pattern that they were seeing. And so they took a really simple approach. They took 10 tumor suppressor genes. Again, these are run-of-the-mill genes, nothing particularly out of left field, your BRCA1s, your PALB2s, RAD51. And they did adaptive sampling, and they did some confirmatory work using targeted cDNA sequencing. And what they were able to find was that for these families who previously didn't have any answers, they were able to find these deep intronic variants. And again, that is a value of adaptive sampling. You're not sequencing just the exons, you're sequencing end to end the promoter, the introns, the exons, the full length of the genome. And so they were able not only to find these deep intronic variants, they were able then to show using long read cDNA sequencing that those variants were causing a pseudo exon and that was leading to a truncation and therefore an insufficiency of that particular gene. And they were able to see that using the long read cDNA sequencing. And importantly, they were able to find some of these variants across multiple families. So maybe they weren't as rare as one may think. These were two families who were unrelated that had the exact same variant. Using some lineage tracing, they were able to find that these families were both coming from Sicily and maybe had shared a relative in some sort of founder mutation long ago. But again, maybe these deep intronic variants aren't as rare as we think. And importantly, these were, again, canonical genes that are thought of in hereditary cancer. And so I think the more we do these type of deep intronic sequencing, the more we'll understand that we don't know what we don't know, and the value, again, of getting these full-length sequences for each gene using adaptive sampling. And so in this particular study, the authors were able to find 92 rare or private deep intronic variants in 88 of the families that they are looking to validate further currently. And so I hope we've convinced you today that not only adaptive sampling has this flexibility and ability to batch, change the content of your beta file, and have been optimized to really kind of fit for purpose for your needs. Miguel has done an excellent job really doing a deep dive into the different steps from DNA extraction to library preparation, what happens on the sequencer when it's making these decisions for target enrichment, and highlight a bit of our Epi2Me workflows for analysis, both out of the box versus the custom workflows through human variation that you can really leverage to answer your unique questions. And so we have a suite of optimized adaptive sampling workflows, our out-of-the-box solutions with the hereditary cancer panel and the pharmacogenomics panel, and then as well as custom enrichment capabilities using our human variation workflow. And I invite you again to click to scan the QR codes on the screen and learn more about each of these different applications. So thank you for your time. And a quick final disclaimer, and that is that Oxford Nanopore products are for research use only. You can also find more information about today's broadcast using the QR codes that were displayed on the screens, This webinar, along with all of our previous podcasts, are also available for you to watch on demand. So please visit the Oxford Nanopore website to check them out. You can also find more information about our upcoming webinars and knowledge exchanges on the Nanopore website. And lastly, if you have any further questions about your project or getting started with nanopore sequencing, please contact support@nanoporetech.com. Thank you all once again, and please enjoy the rest of the day from wherever in the world you are joining us. Thank you.